Hi folks, welcome to Lab 17. This is our second fungus lab where we will be focusing on molds as opposed to yeasts. Molds are the multicellular and in some cases macroscopic forms of fungus. And there's not a whole lot that I can say to introduce this lab because this is a lab where you are going to be tasked with solving a mystery, which is what species of mold have you been provided? There are 8 different species of molds that are the possible species that you will be provided, and all of those are outlined in your lab activity, so make sure you thoroughly read the background of your lab activity. What the procedures for this lab look like would be you would be provided with a unknown species of mold in the form of a tube, and you would take 2 drops of that unknown species and add it to the center of a Saburo agar plate. This type of agar is specifically used to cultivate fungus, especially molds. And after allowing it to incubate for a number of days, each of these 2 unknowns would blossom into a plate that looks something like one of these 2 here. And then you would be tasked with taking a sample of the spores, which are now present on the plate, and observing them under the microscope. And so what you will be provided with in terms of results are the appearances of the spores under the microscopes and a picture of the plate as well, the growth on the plate similar to what you see here. And between the appearance of the spores and the appearance of the growth on the plate, by referencing back to the introduction section in your lab activity, your task is to figure out which 2 unknowns you have been presented with. There is a system in the results section to determine which 2 unknowns you should use, and it's based on the first letter of your last name. So for example, if the first letter of your last name is letter A, you'll be using a certain 2 unknowns, but if the first letter of your last name is the letter S, you'll be using a different 2 unknowns. So make sure you just pay attention to that when you are deciding which results you are tasked with interpreting.